Okay, I wanted to put together two last minute videos to kind of give you an overview and maybe a tool that'll help you study these things. So I've picked up the pictures that are relevant to the T cell receptor pathways that we are going to cover on the exam. And I've got a second presentation that has the same graphical version of the B cell receptor signaling. So that's what this is. I'll just hit the high points here. The details are in the chapter and in the other lecture. I'm looking for a little bit more than what I'm giving right here, but I want to make sure that we get it all kind of in order. That's my goal here. All right, first of all, what are the parts of the T cell receptor and its associated proteins? So you got your alpha and beta chain of the T cell receptor, fine. You got your zeta chains, which sit there right underneath the T cell receptor and interact with it. You've got your CD3, which is actually four proteins, an epsilon and a delta, and a gamma and an epsilon. And these yellow boxes are ITAMs. So these are immune system associated tyrosine, and they are going to be activation motifs. So when we put phosphates on these tyrosines, we are going to activate these sites, and we can then bind proteins to them that will become activated. It's like a universal on switch that other proteins can run up against. All right, so what do we do with this? Well, we attach either a CD4 or a CD8 to it, or else it doesn't work. Yeah. And the reason why we have that there is because hooked to the tail of the CD4 or CD8 is going to be the protein called LUC, which is a kinase. And so you can see this here. These are all the parts of it. And so CD45 will take off that phosphate and activate it. CSK will add the phosphate and shut it back down. And then once LUC is activated, it will phosphorylate the ITAMs on the CD3 and on the zeta chains. And it's the ones on the zeta chains that are going to be more interesting and important for us, I would say. So the next thing that comes along is ZAP70. And so ZAP70 has two SH2 domains that are kind of blocking the kinase from working. They're kind of stretching it so it can't move. And as soon as we have two phosphotyrosines in an ITAM for this to bind, it will bind. That'll open up ZAP70, and ZAP70 can then go off and phosphorylate just about everything else. So here are ZAP70s associated with CD3 and with the zeta chains. It was named because of the association with the zeta chain, but it can attach to these other ITAMs as well. All right, then on to the next slide. What does this all lead to? Well, before we go into that, at the same time as we have activation through all these, so that's the T cell receptor itself, we've got to have co-stimulation. And so the antigen presenting cell needs to present something like a B7 protein made of B7.1 and B7.2, and that's going to interact with CD28 on the T cell side. The MHC and the B7 are going to be on the antigen presenting cell. That will have binding will cause phosphorylation to happen in the tail of CD28. Once there is phosphorylation in the tail, then PI3 kinase can attach to the tail, which puts it conveniently right near the membrane. And so PI3 kinase will take PIP2 and turn it into PIP3. That's what it does. So imagine this compound with two extra little phosphates here, going to this one with three little phosphates, including the one on the side. All right, once we have PIP3s serve as a carrying platform, like a vehicle for a variety of things. So we are not going to cover the PDK and AKT pathway in the test. It's just part of the graphic here. We are going to cover, though, this one and this one. So here we've got ITK, which is going to be activated when it binds to PIP3. And PIP3 is, of course, only made when we get this co-stimulatory signal. And then, let's see, the ZAP70 has meanwhile put the phosphates onto SLP76 and LAT so that these things are all ready to bind other things. ITK binds to SLP76, phosphorylates PLC gamma, which is now activated. PLC gamma is, of course, also attached to one of those PIP3 molecules. And then the other thing that can happen is that our PIP3 will actually bind a VAV protein. VAV protein will also interact with this same complex. So this is GADS in the middle, SLP76 on the side, and LAT running along here. So there's LAT, GADS, SLP76. And I would say practice drawing these things out. Just sketch them. It doesn't have to be beautiful. It just, the shapes and the colors are meant to help remind you of what it is that we are talking about, basically. The result is that VAV is going to open up inactive CDC42, get rid of the GDP that was in here, insert a GTP into CDC42, and then CDC42 is going to go out and start modifying cytoskeleton. Which I think is going to be in the next one. There's our CDC42. So there's the pathway we were picking up. Inactive CDC42 turns into active CDC42, which will then bind to active WASP and NIC, and active WASP will then pick up ARP2 or 3 and whip, and will actually start polymerizing actin molecules. So that is the way that the cell starts to move. This is like, what happens after you turn the key in the car and you begin to drive. So we're now fired up. Okay, let's go back. So we covered this and we left off there with our ZAP70. So now ZAP70 is going to come in and it phosphorylates LAT and SLP76. And so I'm going to move this slide, I think, earlier. That allows us to form this complex with LAT, GADS holding on there, and SLP76. And then PIP3s will also be made because of the co-stimulatory molecule. Those will be floating around at the top. I wish there was one graphic that had all of this on it. I feel like that would be easier, but that graphic does not exist, at least not in the book. So yeah, you got to make one in your notes maybe. Anyway, yeah. Then we've got this part that we already talked about, ITK binding, phosphorylating PLC gamma. And then once PLC gamma is phosphorylated, it can then begin to do its work. And so phosphorylated PLC gamma is going to take PIP2, and it's going to turn it into diacylglycerol and IP3. I think I said it takes PIP3 at one point. That's incorrect. It takes PIP2. Yeah. Let's see. And these two are going to go off and do different things. IP3 is going to go down here, open this ion channel, let all the calcium out of the ER lumen and into the cytoplasm. Once the calcium is in the cytoplasm, it is going to bind to calmodulin, which causes it to open out. And then opened calmodulin can bind calcineurin. Calcineurin can take the phosphates off of NFAT, and with the phosphates off, NFAT can go to the nucleus and actually activate gene transcription. So that's one little pathway right there. The other thing that can happen is that diacylglycerol can go off and we can do diacylglycerol signaling. So here is the diacylglycerol signaling. It is going to bind to PKC theta, protein kinase C theta, which is going to phosphorylate karma. You're going to get a couple of karmas together. They're going to make a scaffold with BCL10 and MALT1. They're old friends. So yeah, just hanging out with BCL10 and some MALT. TRAF6 down here at the bottom is going to be a ubiquitin ligase. That's going to be attracted to our lovely karma platform. And TRAF6 is going to, do its thing on the karma platform, which means it's going to make a polyubiquitin chain, which is going to attract TAC1, and there's tab 1 and 2 associated, but TAC1, that's the important bit. TAC1 is then going to activate IKK. IKK is going to activate I-kappa B. I-kappa B is going to be degraded, and NF-kappa B can then go to the nucleus and induce transcription. So that part, last part's familiar, I think. All right, so we followed our diacylglycerol through, we followed our IP3 molecule through, and that is going to be the two sides. So you are either going to tell me about everything on this page. So the T cell receptor complex, blah, 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 how we activate everything, and assemble these structures over here. This lat, slip76, and GADS, this complex. There we go, complex. And you're also going to mention how we're going to turn all the PIP2 into PIP3, or at least some of it into PIP3. And then you're going to have mention that the PLC gamma is going to cut the leftover PIP2s. And once it's cut them, then you're going to tell me one of the things that happens. So either we're going to have the CDC42 pathway, and the CDC42 pathway is going to lead over here to polymerization of actin, or we're going to have the diacylglycerol pathway, where we're going to eventually make NF-kappa B in the nucleus doing transcription, or we're going to have the head group, the IP3, which is then just going to open up those calcium channels, let the calcium out, and let calmodulin do its thing to calcineurin, which does its thing to NFAT, which then gives you NFAT transcription. So NFAT transcription is going to be one outcome, and you're going to trace it back. Actin polymerization is going to be another outcome, and NF-kappa B is going to be the third outcome. And I might set it up so you have to trace two of those back. That's a pretty good coverage, I think, of the topic. All right, and that's it for T-cell receptors. So I hope that helps studying, and I will put the PowerPoint up there so you can use that as well.